Hello and welcome to yet another snippet from Palebluethoughts. This week we will discuss an incident which caused a lot of flutter in the media recently. The media and the social media were abuzz with the news that one of the ministers who participated in the vaccine trial was tested positive for COVID-19. Anil Vij, Home Minister of Haryana, tested positive for COVID-19 just days after he participated in the third phase trial of a vaccine called COVAXIN. This has resulted in many people raising doubts about the efficacy of the vaccine, and anti-vaxxers or people who are against vaccination are out brandishing their rusted swords. This news comes at a time when the whole world is eagerly waiting for a vaccine so that we can resume our lives in a normal manner. When I spoke with some of my friends and family and also saw some posts and videos on social media, I felt that many people do not understand how vaccine trials work and sport many misconceptions. Hence we decided to make this video in order to clear some basic doubts. We have done a video on immunity boosting and how vaccines work previously, and you can click on the card that you would be seeing above to watch that. The vaccine in question, COVAXIN, is a vaccine for COVID-19 being developed by a company named Bharat Biotech in collaboration with the Indian Council of Medical Research or ICMR and National Institute of Virology. The indigenous inactivated vaccine is developed and manufactured in Bharat Biotech's BSL-3 or Biosafety Level 3 high containment facility. The vaccine received DCGI approval for Phase 1 and Phase 2 human clinical trials, and the trials commenced across India from July 2020. After successful completion of the interim analysis from the Phase 1 and 2 clinical trials of COVAXIN, Bharat Biotech received DCGI approval for Phase 3 clinical trials in 26,000 patients in over 25 centers across India around mid-November. Anil Vij participated in one of such Phase 3 clinical trials in Haryana. The company, in their product description, mentions that 2 shots or intermuscular injections are required, which are spaced some 28 days apart. The literature also mentions that Phase 3 trial would be a double-blinded, placebo-controlled, randomized controlled trial. So what is this placebo-controlled, double-blinded, randomized controlled trial? Let us break each word and look at each one in detail. A randomized controlled trial or RCT is a type of medical experiment that aims to reduce certain sources of bias when testing the effectiveness of new treatments and vaccines. This is accomplished by randomly allocating subjects mostly into 2 groups called the control and the experimental groups, and then comparing them with respect to a measured response. The group called the experimental group receives the medicine or vaccine being trialed, while the other, the control group, receives an alternative treatment such as a placebo like saline or fake pills and not the actual medicine or vaccine. The groups are monitored under conditions of the trial design to determine the effectiveness of the experimental intervention, and efficacy is assessed in comparison to the control group. Please note that there may be more than one treatment group or more than one control group. It all depends on the design of the trial. This is not a new thing in science. RCT has been in use since 1747 when Dr. James Lind conducted such a trial among patients who had been infected with a disease called scurvy. Lind, through his controlled trial experiments, found out that the citrus fruits can cure scurvy, thus leading to the identification of vitamin C, a concept that was unknown at that time. Even today, almost all medicines that reach the market go through randomized controlled trials to find out their efficiency and efficacy. Now, how do we randomize the trial? The patients are segregated into groups, usually done by a computer using random chance. This eliminates any selection bias and the group is completely random. So, what is a placebo? A placebo is a substance or treatment which is designed to have no therapeutic or medicinal value. Common placebos include inert tablets like sugar pills, inert injections like saline, etc. In general, placebos can affect how patients perceive their condition and encourage the body's chemical processes for relieving pain and a few other symptoms, but have no impact on the disease itself. A placebo effect is a scientifically proven influence where your own brain believes that a medicine or a treatment is effective when it really is not. Placebos have been known to be objective, such as resulting in low blood pressure, or subjective, such as a reduced perception of pain. Placebos have known to work even when people know that they have been given a fake medicine. Placebo involves a complex neurobiological reaction that includes everything from increasing the feel-good neurotransmitters like endorphins and dopamine. All of it can have therapeutic benefit. The placebo effect is a way for your brain to tell the body what it needs to feel better. However, the crucial point is that placebos may make you feel better, but they will not cure you. Now coming to double-blinded. Most clinical trials are usually double-blinded, meaning that information which may influence the participants is withheld until after the experiment is complete. This means that the people who participate in the trial as part of one of the groups and the people who do the testing do not know who has received the actual medicine and who has received an alternative treatment. Why do we do this? We do this because effective blinding may reduce or eliminate some sources of experimental bias. Suppose you know that you have been given a medicine. Your brain would start to release the feel-good chemical messengers or neurotransmitters, and you start to have the placebo effect. The reason why doctors are also blinded is to eliminate them passing on the information about the medicine being real or fake to the patients. So, in a double-blinded trial, the patient does not know if they received a real medicine, and the doctor also doesn't know whether they are giving a real or a fake medicine. A well-blinded RCT is often considered the gold standard for clinical trials. Blinded RCTs are commonly used to test the efficacy of medical interventions and may additionally provide information about adverse effects such as drug reactions. Now let's put them all together. A randomized double-blinded placebo-controlled trial effectively means a trial where the people are chosen at random, then randomly segregated into two groups, and one group gets the actual medicine or vaccine and the other group gets a fake medicine or no medicine. Both the receiver and the giver are not aware of who received the actual medicine or vaccine and who received the fake medicine or vaccine. All this is done to ensure that we eliminate any sort of bias or predisposition within the trial. With me so far? Once the trial phase is over, the researcher looks at all the symptoms, side effects of both the groups and compare it against each other. If they both have the same reaction, improvement or not, the drug is deemed not to work. The experimental group or the group which actually got the medicine has to show a sufficiently higher level of effectiveness before we can say that the drug or vaccine works. Thus, we are effectively eliminating any placebo or ensuring that the medicine actually works and it is not your brain trying to tell you that you are all right. Okay, end of story? No, the trial doesn't end here. The results of the trial is published in a scientific journal or sent to a group of people who are considered experts in that field of medicine, and they are asked to review the study starting from scratch. This is called a peer review. They pore over each minute details of the study and look at all the minor aspects of the study, such as the test design, dose of the medicine, side effects, efficacy and efficiency, and then conclude whether this trial can be considered as a valid study or not. If the vaccine or medicine passes through all these steps, that is when it can be sold in markets. Now let us come to the situation of the minister who tested positive after taking the first dose of the vaccine. As mentioned earlier, this one news can result in sowing seeds of doubts in the minds of common people regarding the effectiveness of the vaccine, and people may end up not taking the vaccine when it is available after believing these news reports. So let us see what we can decipher from this news. First, the minister participated in the third phase of the trial, which was a randomized controlled trial. And we don't know which group he was in, whether he was in the control group or in the experimental group. If he was in the control group, then he wouldn't have received the actual vaccine. Secondly, he had taken only one of the 2 doses as prescribed by the vaccine manufacturer. So just by taking 1 dose of vaccine, you cannot prevent the coronavirus from infecting you. Thirdly, we don't know if the minister was already infected with the virus when he went for the trial. Usually, the symptoms take a few days to appear, or a person can be asymptomatic, as we have seen in many cases. So just because the minister attended a vaccine trial doesn't mean that he was safe from the virus due to the 3 reasons mentioned. Now there are 2 things to consider here. We need to identify a medicine's efficacy and efficiency. Efficacy means a study of whether the vaccine works within the body and how does this work within the body. Efficiency study means that we know a medicine or vaccine works. However, we need to find out percentage of success. The current COVID-19 vaccines which are in trial have various efficacy and efficiency values. The German-made Pfizer vaccine demonstrated an efficacy rate of 95%. Moderna vaccines claimed 94%, and the Oxford vaccine at 70%. All these values are higher than the WHO stipulated efficiency value of 50%. So we hope that you now have a better understanding of how vaccine trials are conducted, and you have an understanding why it is too early to come to a conclusion about the efficacy or efficiency of the vaccine, as they are all under the trial phases. There are more studies to be conducted before they are deployed. Till then, let us stay away from any misleading news that may be spread by anti-vaxxers and people who may have been misinformed. We all should have no doubts that only a vaccine can stop this virus. Let us keep our fingers crossed for the effective vaccine to reach the market so that we can say goodbye to COVID-19 forever.